my name's Richard Farragher. I'm from the Knox Environment Society, and we'd like to welcome you here this morning. It's great to see you all here, excited and ready to rock and roll. And you're all welcome here at any time. So if you come in any time and identify yourself from one of the other nurseries or friends groups, you are always welcome. There's always tea, coffee and chocolate available because that's what we work on. So we can't pay money, but we can pay in food and drink. So thank you for coming on a Saturday morning. I know it's hard, but it's always hard trying to find a time for busy people to get together. And I know that everyone's busy, but I thank you for coming. And obviously we're coming into a busy time of the year. Not that there isn't a busy time of the year. Because people say to me, "Are you busy at the nursery?" I just roll my eyes and think we're always busy at the nursery. But yeah, with seed collecting, it is a really important time for us. So just a quick introduction. Obviously seed collecting is central to our operation. We can't grow large numbers of plants without them. So having enough of what you want and need is always a balancing act. In here in Knox, for example, and I know we're a little bit different from, say, some areas that have got large areas of bush, but for us we have 185 plant species, or 41% of all Knox's surviving indigenous plant species are critically endangered. That means they're right on the brink of disappearing altogether. Okay, and then if we put together all the others that are locally threatened, it comes to 84% of our local flora is in danger. Right, so they're endangered, vulnerable or critically endangered. So we have to really think about our seed collecting. A lot of these plants are genetically isolated, and so just going to one spot and collecting seed from one group of plants is not doing those plants any favour. And even if you're, from out in the Yarra Ranges, if you just collect seed for a particular species from one site, and you go there every year and just collect seed from that site, and you're growing 1,000 a year, and they're going out into the populace, you're not doing it any favours because you're actually lowering the gene pool rather than really increasing it. So we have to think about that really carefully as future people involved in this industry. Okay, and as you be aware, we're getting more and more people wanting to buy more and more plants, so the pressure's on. So we have to try and do the best that we possibly can. So you can see here for Knox, it's not looking good, because all of our sites of biological significance are in trouble. And they're all got species that can disappear. So what we're trying to do is trying to push Knox City Council to do something about it. Not just plant willy-nilly, because we know that willy-nillys are no good. They need some sort of organised plan. So care and protection of the environment is important, and in our seed collecting activities we should ensure that we minimise damage while maximising production. So collecting in high yield years and bagging seed so you're only getting ripe seed. So you'll notice that this year some things are flowering really well and seeding really well. So make hay while the sun shines, and if other things aren't doing so well, maybe wait, if possible, collect what you need, but then wait, if there's a good year, again go really hard. Okay, and then maybe you don't collect that again for another 10 years, or you produce stock pots and collect from them. I'm sure Jamin will talk about that. Yeah, so that's sort of just I want to do as a bit of introduction, just to make sure we're continually thinking about our responsibility and our role here. So that's why we thought at this time of the year, just before we all head out with our little seed collecting bags and all that sort of stuff, that we try and maximise what we've got to do. So we've got our guru, the man who can talk in wet cement, Jamin.
If anyone's on a stool, you're in trouble. I'm going to talk that off.
So Jamin's worked at a number of nurseries, and people probably may well know him. He knows more about plants and stuff than, it's just unbelievable. So it's good to have him talk. So I welcome Jamin and thank him very much for his time this morning, and the floor is yours.
What I want to talk about to start with is when you're starting seed collecting for this season, your starting time is back when the season starts, which is back in August. when the first things start to ripen, which are Hakeas, Banksias, and the Proteaceae family. So you're starting then, and that's where your planning starts. So you know where these plants are, you're going out and you're recceing other things that are there going, hey, this year, for instance, this year the acacias were the most fantastic flowering I've seen for years, probably 10, 15 years I've seen. This has been the best year. Whether the seed's going to produce to live up to those expectations, I don't know at the moment. I've gone back out and had a look at a couple of them, so I go back to these sites. It's actually what my job here is this year, is recceing for them to seed collect. So one of our plants is Acacia acylatissimus, which is really rare, and it had a magnificent flowering year this year, really nice growth. I've gone back and looked at the plants, not much seed production. We've got a couple of pods here and there. A lot of the pods had gall on them, and I'll show you some of the gall. They've got this, it's like a little square, it's stopped. So unfortunately for them, but others are looking really good. At home I've got Acacia suaveolens down my way, and it's produced magnificent seed this year and has flowered well. So it's not for every species that has missed out, so you've still got it. So what you do first is you need to be recceing. You need to be going out to these reserves, seeing where these plants are, what you need to collect, what you need and what you don't need, and where you get into the place. Really important thing with reserves, especially most of us here, other than Yarra Ranges and that area where you just drive into a massive place. If you've got a little reserve, what's the best entrance to go from? Which side? So you know, if I'm going out today and I'm going to collect seed in 5 different reserves, what's my plan to get around the 5? Do I write up a plan? What I'd normally do, sorry I don't do PowerPoint presentations, so I'm sorry there's nothing you can look up on the screen. You have to look at me all the time. What I normally do is I'll look at a species or a couple of species I'm collecting that day. I know, for instance, I'm going after Platylobum obtuse angulum. I'll look at the reserves they're in. I can go here, 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 basically in a circuit. It's basically like delivery. A delivery truck driver would go, I'm going to start here, end up here and come back to the pack home. And I'll have an approximate time that it might take to get to the place, and I'll be there. So it might take me 15 minutes to get to one reserve. I've got 10 minutes, so I'll have 15 minutes in there. So I've got to be at the next reserve at this time, and I'll have priority numbers next to these things that I'm collecting. And while I'm there, I'm looking at other stuff. If there's something else there that I really need while I'm there, I'll collect it as well. And I'll go through my list as I'm running around. It's horrible 30-degree day, you're sweating, you stop at the milk bar. I used to get an icy pole at the milk bar there when I was in Croydon North here, stop there. But you go around, you'll find that, geez, this reserve's only got this in it. I'm 20 minutes over time at the moment if I'm going to get back. So I'm going to avoid that one and go to the next one. So that's bit of work you do when you're doing recceing. Know where you're going, and what I was saying with where you're getting in, when you're driving around, you're doing this planning. If there's 2 entrances and the other entrance you have to walk 50, 100 metres to get into the reserve, maybe it's better to get up to the other entrance so you're in and out, especially if you're doing it on a hot day. You've got a limited amount of time to collect some of this stuff on those hot days. It drops really quick. You might have, oh shit, the Pulteney Gunny Eye's dropping today. I've got to get there. And it will drop in that day. You might be getting in the morning and you've walked 100, 200 metres in the morning and wasted 20, 30 minutes, and you get to the last one, you go, it's dropped. If I was here 30 minutes earlier before I would have got it. So there's crucial things like that to do, to plan for yourself, where you're going to go and collect it, what you're going to collect, and how you're going to collect it and store it. Richard, just a couple of ways. Richard carries his nice man bag around. I normally carry a shopping bag, and I'll have my, or even I used to have a fold out, those manila fold outs, and I just have my seed bags in them. I just pull out that one and go to that one, yeah, that one, and put them back in. I'll just carry that in, or a bag. One of the green Safeway bags are really good, and you just have your bags in them. You get them from there. When you get back to the car, first thing you do is you label everything you've collected from that reserve. Okay. Something I didn't put at the start, sorry, I forgot this. What this talk is more about is practical things that you need to do to collect seed. I will talk about some of the plants and ways to do them so you learn that, but I'm not going to go through cleaning and a few of the other things that Richard's put up there. So I'm sorry I've cut that back in. But you want to make sure that when you get back to the car and you've got 5 bags, you've got Dilwini, you've got Platylobum, you've got whatever, you've got some Dianella revoluta and whatever is about that time, you've got them, label them there, even if you just put where they're from. The other thing you need to put in bags are signs, and signs are part of the plant, whether it be eucalypt pods, whether it be a part of any of the plant, bit of the glooms off stiper. So when you collect the stiper, make sure you put the glooms in as well. So you're sure it's that species. Some of the other places, I mean, Yarra Range has probably got 10 species. We've got 3 here, and they're very easy to distinguish just by the glooms and the time they get collected. So if someone's collected in November, I can already go, they're pubinotus, because Rudis and Australis are not November. So those type of things are really important. When they get back to the seed cleaner, whoever's cleaned it, they go, well, what did you do there? Getting onto that, the next part of that spit is when you're collecting, writing it out. So even if you just put where it's from, when you get back, you know what the species in, put the species in properly, the amount of plants that you collected off, okay, what you collect off, they're really important, because in future years, if you've got that seed stored away and you go back to that reserve and there were 6 5 years ago and you go back then there's 1, you know they're in trouble. You're going, hang on, we've got to do something about this plant. This becomes a priority that we need to start building its numbers up, regrowing. So we also have the date, obviously, collector's name. Got to have your name on it. If I get a bag here and I've got no idea who's collected, I can't ask them, where did you get that? Was it remnant? The other thing we put it whether it's remnant or planted. A lot of planted stuff, especially in reserves, council reserves, can be grown from small gene pool to start with. You might see acacia stricta in a reserve, go, oh, I'll collect all this, it's all planted. But it might have only been collected off 1 or 2 plants, which is inbred. And it might have been inbred for years and years. This is really weak seed. We're looking for stronger seed. The problem we have now that you might have acacia stricta in a reserve and there's only 2 left there. You either get that seed or you go back the generation and take the cuttings, grow the plants, have some from somewhere else, cross-pollinate, and then collect your seed, so your gene pool's much higher. So we're looking at things like that too, so know what you're collecting when you're going in there. If you've been in there, if you go into a place and there's 10 plants and you've been in there 1 year, especially with Fabiaceae, I would collect Fabiaceae 1 season, and if I only got a gram of something, say it was Hovia, next year there was heaps of there, I would just add it to the season before. If you sowed it, if you had 1 gram and 6 grams, you'd probably sow the 1 gram and half of the 6 anyway. So why not just put it together? It doesn't really, as long as you've got the early date on it, Fabiaceae, season to season, doesn't matter. But when we're talking about grasses and other things, make sure it's separated, because some years grasses can be good, some years they can be Prostranthera. There's a beauty. That one, you can get a great year, seeds every year, go and collect it, none of it comes up. You can get another year, we got some this year, and it all comes up. How did that happen? So make sure you separate your seed into years, and the collector, as I was saying, the collector is really important to know who that person was, where did you get it, what time of year. And we're going on, that's the next one we're going to go on to, is timing and ripeness of seed. Collecting seed too early or too late, really bad. Go with the early stuff first. Not ripened properly. I've got the Hovia there. It's green. Nowhere near ready. Same with the Canetia here. Still hasn't turned into a peanut where you can break this pod open and see the black seed. The green seed on those Hovias, nowhere near it. I'm going to talk about bagging later and show you how to bag, but I wouldn't bag that at the moment because it's still too green. That Hovia. Hovia's a bit, some of the Fabiaceae are really sensitive to being bagged. Hardenbergia is another one. If you're going to bag Hardenbergia, which I would suggest you don't, because it usually hangs on. It's really easy to collect. Putting bags on only just hampers the way the seed ripens. And same with Hovia. But Hovia, the reason you do Hovia, you'd bag, we'll talk about bagging later, but I'll just put this in now. It's part of the collective. The reason you bag some things is so you can find the bloody things again. Really hard to find Hovia late in the season. Go, well, where the hell was that? You might see a couple of black seeds and walk past that magnificent one that's fallen over under the grass and never see it again. Go, oh, we could have got that this year and then grown them. One perfect example is Wormbia. Wormbia is a shocker. You'll see the thing flowering and you think, I'll come back and get that. You will not find the seed pods at all. They don't like to be bagged when they're green, but if you do bag them early, like if you bagged one earlier with a lighter colour bag, you will be able to find where they are and then recollect it later on to get some seed to give you a start on them. Easy to grow. They come up easy in these pots. So, but once you've got your stock of them, which I'll talk about later about having stock plants, then you can grow from your own plants and not have to go and raid these places anymore. Our main aim here is to build our gene pool like Richard was talking about, and then we're collecting off our own plants, and then we're upping the gene pool in the reserves, not doing it the other way. We want to put back, not take. So we start off, build our own numbers, build the gene pool up, and maybe you're only doing 1 or 2 species at a time doing this, but it's the way to go, growing in pots. So we go on to, if we go back to the ripeness, so I was talking about ripe seed, collecting it under ripe. If you bag it too early, I've bagged Hovia like that because I think I'm not going to get back there. You know, I'll just put it on now. The seed came out, and I've got some Harden Berger here I'll pass around. That was collected way too early. Sorry, I'll get it out a little bit later. We'll do that a little bit later. But collecting it too early, the seed's weak. It doesn't ripen properly. It doesn't actually form a proper seed, and it doesn't come up. I collected some years ago up on the pipeline track, and we went up there, up in Mount Evelyn, and we thought, we'll just bag these ones on the cliff here. They never grow anyway because they mow it all, so it never grows. So the seed's wasted every year. So we'll just bag it. We're not going to get back here. It was November, late November. We were taking cuttings up there, and I thought, we're not going to get back here. I'll come back and get it. Went back there in December, January. Seed was garbage. I put it in, didn't come up at all. So if you're going to do it, put your effort in it. If there's a plant you really want to collect, aim at that. Don't go off, I'm going to get a bit of that and a bit of that. I'll come here, I spend an hour, I get half a dozen seeds, I'll come back in 2 days, I hit the jackpot, I've collected it. If you go back and back and back to a place and aim at one or two plants to aim at, you'll get them. If you just go around the places and try and get bits and pieces everywhere, that's what you're going to have. And you're going to have these little amounts in your seed bank of 0.5 gram, 0.1 gram, and then when you go to put it in, if something goes wrong, it's gone. So you want to build your numbers up as well before you do that. Now, one of the things on the seed collecting, I'll pass these around. My helpful helper here, Peggy, is going to pass these around. I want you to have a look at these. Now, when you go to reserve, you'll see things on the ground. There's obviously ukes. You'll know what species are in there. I just want you to have a look at these just to, I'll ask questions about them later. Just take note of both of them. So there's 2 there, you're going to hand them around. When you're there, noticing what's there, what plants grow there. The reason I'm going to hand this one around, some of you might know which species it is straight away. They are. But anyway, if you don't, just have a look at what they look like, and we'll do that anyway when they get around to you. But knowing what species of eucalypt are there usually can give away what plants are there as well. If you go into reserve and, geez, there's messmate everywhere here, there's a good chance there's orchids, there's lilies, and there's everything in there. If you go into somewhere where there's radiata, like Bungaluk for instance, which is Cephalocarpa and radiata, it's a wet, heathy forest, grassy forest, so you know what you're going to expect to find in there. So if you already know what plants are in there, and you can use that knowledge, if you go somewhere you haven't been before and go, oh, look at these plants are here. This plant might be here, or it's a perennial one, it might come up later. I'm going to come and look for that plant there because it's in this area where these plants grow the same there. So obviously we have different EVCs. I don't want to give away what they are. I probably could, but where they come from, the certain plants that grow that you usually associate with that. We'll go back to overripe. Now we've found with stuff that's left on the plants too long, that's collected later on. For instance, any of the Cassinias or the Cassinias, or I'll put this one up here so you can see it. Cassinias or Ozothamnus, that if you collect it at the right time, you'll get seed. Hope you can see that. And if you collect it too late, you get the bracts. There's no seed in that. What happens with the Cassinias, the Ozothamnus, and most of the Ollerias is they have a little bract that holds them. They're like blooms that hold the seed, and it opens a little bit, and it like dust. That's when you collect it. If stuff's fallen straight in the bag, you've probably missed the seed, and you're collecting glue, and it doesn't grow. So that's one. The period is about a week or 2 at the most, maybe 3 or 4 days actually, depending on the weather. Your weather as well makes a big difference to your collecting. Apacris are a shocker. Apacris can be green, looking ripe, going, well, that's green, I've got a few days, and you have 2 hot days. You go back there, and the whole lot's gone. It's brown gone. Hang on, it was green. They don't change to black like the hovias will. They just do it. And you can't really bag it because it has to be ripe. A few of the other plants that have to be ripe, like something like comosperma. If you collect comosperma unripe, it won't germinate, just doesn't grow. But if you get it ripe, I've had volubile come up really well, but it's one of those ones that I was saying, you've got to go back. You go back and you get the ripe stuff and come back another day. You'll get the result, and the result you'll get it is worth you doing it. Whereas just grabbing bits and pieces, you'll find your results growing plants. I've got 6 comospermas, but I did it real quick and I got back to watch whatever you were on TV or whatever you're doing. But if you do out there and take your time, you'll get the results out of it. It is really worth doing back and forth. The recce's to start off to know where these things are, and then going back to go, that's the plant I want. I know where it is. On that, when you're collecting, we'll add these in. You're making sure you're not trampling 20 other plants to get into whatever you're getting. If you're going in to get something as low like Bossia, it's usually in with other low plants because that's why it's there. When it's on a cliff face and it's doing well, it's easy to get, that's great. But when it's on the ground, you've got to climb in there to get it. You watch where you put your feet. You make sure you're in the moment. Now, I did the pain course after my accident, and the one thing they taught me is I remember, it's worth doing, crunch your hands up and feel it. You're in the moment, you're there. You know you're there. So when I'm there, I know I'm there. I know I'm not thinking about the donuts Richard's cooking us now for you to have later. I'm walking in between and not standing on that hovia, that plant. I'm in between it. I'm over there. I'm standing on an Erhard, or I'm standing on a bloody paspalum or whatever other weed. Stand on them. Use them as your platform. Bugger them. They're there for you. They're there for a reason. So use them. I do. But so when you go out, think about what you're doing before you're collecting. Be in the moment. The other reason to be in the moment is, well, Richard told me snakes. I've never seen it. If you're in the moment, you'll know if the snake's there. If you're in areas like that, all snakes will run away. Even the copperhead. I had a copperhead. I was in Maffra and up the top of Maffra up there in the hills there, and we're walking along these tracks, and of course I was away with the fairies, and I got about from here to the front row away from the thing, just slid underneath the rock. Didn't even bother me. It's a bloody copperhead. You think, oh, they're not. And red bellies are even docile. You probably could walk past it. You'd have to stomp on its head to make it bite you. But if you're in the moment and you're thinking about where you're walking and that, you won't encounter a snake. You'll scare off. Make noise. Clap your hands, sing, we go. And then when I go out with our group, they never shut up. So there's no way anything's anywhere near us. So the other two are bull ants and jump jacks, okay? And leeches. We put leeches in there for whoever's collecting ferns. I tuck my socks into my jeans all the time. I have a second sense with leeches. I know when the damn thing's on me. I can feel, that feels like, there we go, flick it off. I get rid of them, I'm used to them. I know where they are, so you can get rid of them. But bull ants and jump jacks know what their nests look like. The jump jack nest looks like it's like fine gravel over dirt. The bull ant, there's a bull ant nest here. We have a bull ant nest here because we've got healthy soil. That's why we leave them, because they're an indicator that the soil's healthy. So you going out there and you've got your planted area and you've got a bull ant nest there. Oh, we've got to get rid of that. You're doing the wrong thing. Those ants are doing the job that they're supposed to be doing, and they're there because the soil's healthy. So that's why when you're collecting, again, you're up there. I'm collecting Hakea olicina, and it's here, and I've just trampled in and I'm doing this. I've trampled straight on the jump jack nest. They're running up my legs and I'm getting bitten. There's no bracken around to relieve the bloody sting. So anyway, if you get stung by them, new bracken, break it, rub it wherever they bite you, goes instantly. It's like vinegar on stings from octopus and that, what they use with the jellyfish. It's same has the same effect. So make sure where you're putting your feet. Make sure you're not trampling on stuff like that and putting yourself in the, especially if you're allergic to them. I mean, they're on you in no time. I was out on up here yesterday, and they were up there, and where I've been weeding, I got bitten the other day through my jeans. The thing got through my jeans. It hurt. That was a jump jack. So they're buggers. They'll go you. Where the bull ants will come at you, but they're so slow that they're easy to flick off. But the jump jacks, nightmare. So just know what they look like, and if you don't know what their nests look like, look them up. Look up on, we've got Google, look it up and see. So when you go in there, go, I know that's what it is. I'm going around, that's where I've got to collect, but I'm going to come in from this side. So make sure you're doing those precautions before. As I said, in the moment, it's the best way to be, and you know where you are. Think about his donuts while you're writing out what you're doing, and you write donuts instead of some reserve. Donut reserve, what? I've done that. What the hell am I writing?
Have you encountered wasps in the European wasps?
Not really our way, but probably in the suburbs where you are out that way, you're probably going to, because it's probably the only place they've got left, really, isn't it? So where have you done it before? Have you got?
I was cleaning up part of my neighbour's yard. I tossed a branch. Didn't realise there were a friggin' wasp nest there. And they went.
They got you. They chased inside. Yeah, I've had that before. I got bitten by them, but it's usually we don't really see them in the bushland. If they're going to be in the bushland, this little one, their nests are always facing east. They like the morning sun to warm them up. They don't like facing west. So if they're going to be anywhere, and being in that moment, you'll see them doing this. You'll see the numbers going in and out, so you'll be able to avoid them. I don't have any problem with wasps. I don't even see wasps out there. I haven't seen wasps for ages. Really, not out in proper bushland. They just aren't out there. Anyway, we're going. So we've gone through the collecting. Okay, we talked a little bit about you putting, doing the bags, you labelling properly. Make sure you got all that done in the car. You can use different types of bags. Here's a bread bag. These are really good for the bigger seed. Sorry. Yep. Yep.
When you write in the bag, would you please write clearly?
Yep.
Other person's got to read it.
Yes, that's what I'm saying. I always usually carry, if you've got a clipboard, it helps. But a lot of the time, the seed's that far over. Using a texter too, use a black texter. Don't use pen. It just puts through the hole, put a hole in your bag. With bags too, there are good bags and there are bad bags. Okay, you probably know this. Don't go to IGA. Their bags are crap. They fall to bits. So do Safeway ones. The Coles ones are probably the best. They seem to be the only ones that hold together. I went and bought some IGA ones. I was down at Montrose years ago when I was at Candlebark, and I thought, I'll just get them from there. And I got out there, and the first lot I bought fell straight out the bottom of the bag. I lost all the seed. So think about what you're getting. The very bad quality from those. So, the other one, using these magnificent, these Lamandra longifolia, the Poas, straight in there. Now, I prefer to collect Poa running my hand up and running the seed in, because you know it's ripe. But if you have to collect like Ensiformis and Lab, you just cut it off and you put it, because you're just going to use it 1 or 2 years. And I'll talk about time length of using seed. You can do it that way. Lamandra longifolia, you want it, when you're cutting the heads of them, the actual stem turns yellow, and you usually can break it like that. You can bust it with your finger. They're ripe. You can put it in upside down and know, because it's not putting any more, anything more into ripeness seed anymore. They're already done. If the stem's still green, they're not ripe. Okay, if you're taking it, then you're getting seed that's not properly ripened, and you're probably you'll get it to germinate that year. Green seed will germinate that season, but if you dry store it, I guarantee it's not coming up. And then years later, when you actually need it, when that goes wrong, that's going to be the thing. You go, shit, we got to get this, and we're finding that out with a little bit with some of the stuff we've got that's been collected a little bit green. That we're getting to a stage where things weren't collected over the ridiculous time, those 2 years, and we're going back to stuff that when I was originally it was collected that's really old. Unfortunately, we had movement of house, we had problems with our old house with the temperature, and that's probably made the seed go. And I will talk about temperature where you're storing a little bit later. But these bags work really well for the bigger stuff. Thermita, fantastic for thermita. And when you're collecting thermita, one of the things I will talk about is definitely thermita. Collecting it. Normally I won't, someone's who done this, they've cut the heads off. When I collect thermita, I just rip the heads off like that. You get your finger in there, you'll know they're ripe because the awns and the columns spit, they're twisted. And it's around December 8th to December 15th, okay, that timing. And if you know, and with your seed bank, if you've got your proper collection times in, you can always go back and look at those proper collection times to know when you need to go in again. The other thing your seed bank does too, especially if you've got who collected and where, they're there. And with thermita, it doesn't matter. You could collect thermita from anywhere from here to Melbourne, back out this area. It's all the same, very same DNA, very closely related. It doesn't matter. But what matters is if you're able to collect 20 grams from blah blah road, and you keep that down so people in the future know that's the place to go. Because we lose knowledge. Usually with all your groups, I can tell you, every 10 years we probably get a changeover. And a lot of the knowledge gets lost. So one of the things these meetings are for is to pass on knowledge that any of you have back. So anytime, like I got told about, make sure you put the labels on properly. Yeah, make sure you write clearly, please write clearly. Make sure you got a bit of the plant in there. But any input you got back, that's what we want. These sessions are actually to share information. If there's a process of a plant you found works, especially storage. So this mainly, I know I've done collection, but this is mainly about storage. The real problem we're having that we've got in Indigenous plants, we don't know how long seed will store for. No one knows. We know Fabiaceae stuff and the acacias, if you store it properly, probably got a lifespan 100, 200 years. But does all of it come up at once? When you sow Acacia implexa, not all of it comes up. Is there a reason why it all doesn't come up? Is some of it actually grown that it needs 2 fires to come up, or 3 fires, or a certain amount of aging? So we've got plants that we've got things like Platylobium infecundum, Acacia aclatissima we've got in that pot as well. They live for up to 100 years. So they don't want their seed germinating after 5. They want the seed to store in the ground in case something happens to them, and it can still be there for in future years. Even Coronodiums and any of the Asteraceae stuff, I've seen them come up on banks that have been washed away, and there hasn't been those plants around for years. So the seed's obviously stored somehow. And most of the Asteraceae stuff we collect, we know doesn't store very well. But is some of it able to? And they're the keys we've got unlocked here with actual storage time too. I'll back on the Themata. Look, I'll do straight on Themata with actually collecting Themata. I'll get his bag out of the way. I put them in one of these bags, but I will get when they're on the plant, pretend, you can see they're already hanging out there, but pretend they're this high, they're hanging. You run your hand up and you're grabbing all the glooms, all the glooms and the pods like that, and you're throwing that in. You're leaving the stems there. So you run, and you'll know when you run your hand up it, you'll know they're ripe because they'll click off. If you have to rip it off and it rips the stem out of the base of the plant, that's still green. Now the other thing with Themata, they grow in fingers. They do this. So they start, this will be the ripest ones, back to the unripe. And when you collect it, it doesn't matter, but I was saying between about the 8th and the 15th December seems to be Goldilocks time when most of the seeds ripe enough. So that's when you'll get green seed in it, but you will get 2 to 3 ripe seeds that are worth having. The green seed is useless, okay? Absolutely useless. We're talking about early stuff. I did, and I can't show a picture, but years ago I did a test at Candlebark when I was there, and I collected seed. I after ripened it. We'll talk about after ripening with just Themata after this. Sorry, I cut in with that. But I cleaned it up and I thought I'll put the green seed on one side and I'll put the really ripe seed on the other side. So I've collected in December, done this in February. You want to after ripen with all grasses and most plants for at least 6 to 8 weeks before you sow. And this really works with Themata. Themata really works, and this I'll go through Themata properly so you can write this information down because I want you to get Themata right. When I put this in, all of the brown seed, which is this, and pass this around, the really nice dark seed came up. The other one, odds and sods, and I saw a couple of brown ones go on the other side. There was 1 or 2. Nothing else. None of that, even if it was light brown, it didn't come up either at all. So if you're cleaning seed and you're putting in that green crap and all that into this, and then it's got that much and use half of it, well, probably 2/3 of it, it's junk. So you're sowing it and you're probably getting 50 or 60 plants go, I can't grow this properly, we don't do really well with this. How come? It's because your seed's garbage to start with. So when your cleaners are doing it, and a couple of my cleaners are there, wave my cleaners, they're down there. They're our seedy, Ian says, so they're seedy people now. We used to have seedy ladies, but Ian's joined, so they have to be seedy people. So when they do it, I stamp on them. I stamp on their hands if they don't do it properly. They know, Pat knows, that we get rid of the garbage. We offload anything that's not proper looking seed. You do not want to store rubbish seed. It's just not worth doing. It doesn't give you any results. And this is probably one of the reasons why a lot of nurseries have a problem with Themata. we go on to Thermid as well. We talked about the collection time, how to clean it. You know it's got an awn in it, in the back, the column and the awn in the back of the seed. All you do is pull the awn. I'll get it on video here so you can see it. You grab a bit. These are even a bit green, and it just comes out. The awn's out, and we store the seed like that. If it's got any, sometimes it's got a little bit of the old bract on it, the flower bract. It's not actually the gloom because it comes out, and you just rub it off. So if that gets on there, the bit of green, that's a bit of gloom there. I don't know if that's going to be able to get on the thing, but I just rub it in my fingers to get it off so it's all clean. Any organic material you put with seed can break the seed coat down. When you think about it, this has got a coat on it, and how it germinates is by breaking down in nature. And if you leave organic material, rubbish in with your seed, it's not good for your seed. It's going to break your seed down. So you want to make sure your seed's completely clean. Winnowing is very easy with this too. It's heavy enough that you really can't blow it out of one of our bowls. We use the bamboo bowls and the white ones there. It sits in there nicely. And when I'm cleaning it, I use over here. I use the top of this. I don't. If anyone's told you use a towel or fabric, don't bloody do that because you can't. They've got a hook on them. If it goes in, it hooks. You can't get it back out. You've only got to push it through. So don't do. What I do is I grab what I've collected that year. So about after ripening time, because what I want to do is I want to get enough that I can sow, and if I've got enough left over, I'll dry store it. So I'll get it out and I'll put it on my. Even look at that, I even got some Brizza. I've been pulling out Brizza Maxima this week. It's a beauty. Watch that too. When you're collecting this stuff, you're going to get, especially sweet vernal. It always finds its way to sneak itself in. This is why you clean your seed really well. The sweet vernal doesn't have a lemur. It's basically a nude seed which sits in the. Do you know sweet vernal? It looks a bit like if you get it mixed up with Dioxia. I saw some Dioxia yesterday right next to them. I was thinking about grabbing them. I probably should have got them and passed that around and say, hey, this is what they look like. There's actually two forms of sweet vernal. There's the perennial form that's flowering now, and in January, the annual one flowers. So if you've got one seeding and flowering in January and it's a sweet vernal, go after it, get it out because it's an annual, and you can eliminate it. But what I do is I'll go through and I'll just slowly pull bits out, and I'll go through this and I'll grab them and I'll have my container next to me, and I'll just put it in. And it's one of those jobs that our CD ladies like doing. They like sitting down. You get in a big circle and you just talk about stuff and you do that. But you do enough that the after ripening period that you can sow. So the after ripening period is December's, Poas, Stiper, mostly Rigiospermas, or the Rigiospermas a little bit later. That might be December. Definitely Themida and all the other grasses, Dioxias and the Aleocharis and all those. You collect them that time of year, you dry them somewhere. So we want to dry them somewhere where it's airy, warm. We don't want them in our seed house. They're not drying out in there. They've got to be, I hang them like this up in our house up there. If you've got a really hot shed or somewhere like that, you can do it, or your seed house or somewhere where the sprinklers are on that time of year, they can just hang in there, and you leave them in there for those 8 weeks. Then you go through it and decide what you're going to clean and sow that February. And after ripening, seed sowing works a treat. I'm telling you, you'll get your best results from that. Then you dry store what's left over. And that's the stuff you'll sow the next year, coming up now in December. So Poas, I'll sow Poa in early December, late November, early December. So I've got some stuff for that early autumn period when if we get rains early on or you get an early order, you've got some, because the stuff you're doing in February is obviously not going to be ready to May when the proper and for your spring stuff as well. And with Themida, I do the same. I dry store some. When you're dry storing Themida and Stiper, they have a dormancy period. So Themida's is 18 months. I don't know if you've heard. It definitely is. It works. So I've collected it this year. I was going to clean this. Thought I should bring it down and do as part of the talk because I was going to clean it and just dry store it. But this cannot be used until next season. So not now, next year. We can't. This was collected last year. This is not this year's stuff, by the way. So I haven't collected any. There's none ripe at the moment. So again, we're talking about ripe seed. You wait till it's ripe before you go and get it. It's not even ready yet. The only things we've got ready at the moment are Lyria lyrata. If you're into that, that should be there. And we're getting close to Divisia. You could almost bag Divisias if you do that, because I find that's the best way. With Divisia too, you know the seed's ready, rattle it. Go up to the seed and go, hey, if you can hear a rattle, the seeds in, there's good seed in there. It's ready to pick them off before they burst and you lose them. So that's what I do with Thermida. So I'll collect it in early December, and early February we'll go through, and the seed elaters will clean them out for us, and we'll sow that, and the rest will either get cleaned of that then and get put away, or left to the winter months when extra people come in and want something to do. They can clean some Thermida. It's therapeutic, it's easy to do. You only stab yourself 3 or 4 times each. I definitely mouldy. Talking about mouldy seed, I have had stuff go mouldy, and it's actually worked. It's actually worked better. So I've had Pattersonia we collected and left in the bag, and it's a bit mouldy, and last year we looked at it, was April, going, well, what are we going to do? Let's sow it. Came straight up. Usually Pattersonia takes 3 months or so before it decides it's going to come up, and it had been mouldy straight up. Maybe, as I'm saying, this is the information I want to go back and forward. Another one I've done, I was talking about late collecting. Now the two I want to talk about late collecting are Xanthorrhoea is a definite, because it's so good. Oh look, it's still there, it's June, I should get some. The problem with the seed is it's been weathered. It's sat out, it's had the wind, the rain, everything hit it. And you're collecting it. If you're going to sow it that next season, because you really want to sow it, you could sow it then, I guess, but you're going to be out. We normally sow in around early autumn for that, because it usually takes about 15 months in a tube to get to a saleable plant. So there, around the spring next year, there's a plant that's saleable. If you're doing it then, you're probably waiting an extra 3 months. You're probably growing it for 18 months instead of 15. So it's your space. If you've got space, it doesn't matter. You could do it any time. If you haven't, like we're doing here, you've got to juggle. You've got to make sure plants are going at the right time. And if you're collecting stuff like that, you're going to go, well, it's got to go in then next year, or it gets chucked, because it just doesn't store at all. So, and we've got proof of this. We grow Xanthorrhoea that's 5 years old, and we get still pretty good results out of stuff that was collected in February, March, where it's ripe. But stuff that's collected in May, June, July, that time, we found when it's that old, doesn't even come up at all. It's obviously, it's been wet then, it's gone through it's starting the germination process, and you're then dry storing it, you basically killed it, it's died. So I'll show you some Lomandra we've had problems with. We've got some good Lomandra specimens here that hasn't been dried properly, and what comes out. So Xanthorrhoea is one. A couple of years ago, I went out, and this was my main, how I learned it happening. I went out, really needed Joycia pallidum. It was June, it was raining. It was raining on 2 things: don't collect late, don't collect late. I did it. Stupid, but I did it. We were desperate for Joycia pallidum. So there's 2 spots. There was still seed in the pods, the older stuff, not the good stuff that comes out first. So I collected them. I went back to the nursery. I dried it out in the oven. I dried it out so I sowed half of it. It all came up. The beauty, I'm going to store this next, I'm going to sow it in November, December when I like to do it. None of it came up. So the weathering has started the germination process. Me drying it out just gave me a chance to get it pulled it apart, because it was all stuck together and it's still germinated. But then drying it out and putting it away and thinking I'll get away with this, didn't even come up at all. So you think if you're doing that and you're storing it and trying to use it 3 years later, there's no way in the world it's coming up. So you're already behind the 8 ball by doing that type of stuff. So I'd done that one. The other plant I'd got mouldy, and Alice, who's no longer with us, who was our seed cleaner and at both nurseries here at Candlebark, I'd collected these Pimelia axifloras and I'd left them go mouldy, and they were still green, and you're shaking all this mould, dust was coming out and going, oh, there's no way. I was showing her, she's going, what do you think I should do? Oh, just chuck it away, there's no chance. I said, bugger it, I'm going to have a chance. So I took a chance on it. I put it in sand and I put it in the oven and just baked it to just try and dry it out. And I sowed it. Hundreds come up. It worked. So the mould again has made the difference. It's broken down the coat and they've come up. But it's only going to work for that season. So any of these things you're not going to get, I'm going to be able to dry store now and say, hey, 2 years time I can do it. Going back to the Thermida, that's the thing I was saying with the what you want to do is you do your after ripening, you put it in the amount there, in February that you want, the leftover your dry store, 2 years later in that December, you can use it then, or 10 years later. I've found it 7, 8 year old seed that's been collected properly, and it's properly dark seed, germinates. But it doesn't that next season because it's gone into that dormancy. And that's the process of that plant. We don't know plants. We've been here for what, 20,000? They've been here for millions. And I'm not doing the Bible crap. That's not that. I just know that there's no evidence before that. But they've been here for millions. They've been doing this process for how many generations? So they know what they're doing, these plants. We don't. And that's why they do it. That's why they ripe seed. That's why they slowly ripen. So something might happen earlier. They get away with it because later, or the early stuff ripens and something happens later, they've already got some seed out. That's Themata. That's why it's everywhere. It's an opportunistic plant. It does it. It's obviously been brought in with the Aboriginal people from the Polynesians when they came here because it's all through Asia and it's their food source. So why I talk about Themata, because I think it should be planted everywhere. It's gluten-free when they make the bread out of it too. That's what I like about it, and it's not bloody wheat, which has been buggered over and hybridised so much, it's not what it was. So, okay, we're through that. We're back onto those. So I did those pioneers. I got them all to come up. So just remember, you can use some of these other processes. We're going to the Dianella, one that a lot of people have problems with. I know some, was it Keck, who wanted to know about the virus. Daniel's not here. Daniel Flame, and sorry if I CC'd everyone and you got my message that Daniel Flame did and I spelled his name wrong. That's probably why he hasn't come, because I put Flame instead of AIM, and I should know better. Being from bloody Counterbark, I should know better. I get a kick in the arse for that. But he did the study on these, and it's a virus going through the revoluters and that. Okay, and some years they seem to avoid them, and it must have something to do with when they actually grow, with the season, with the weather pattern. A lot of the plants, our plants that pollinate. I don't know if you've got Platylabium infecundum. Some years they will pollinate it. It's got to have something to do with weather conditions and the insect that does it. And from what I've seen, it's the frost that hits those buds at the right time, and it tells the plant that frost's there and that insect's probably hatching at the same time. I'm going to produce flowers that can produce seed. Because I've seen places where it's produced seed before, and in other years it hasn't. And that's been the linking thing. It's been those frosts. There is a little bit around this year, and obviously some plants will always produce. They're all different, so they'll produce it whether they get this, but the majority don't. It's a bit like what Garnia does. Apparently it only seeds once every 1,000 years. Well, if it lives for 1,000 years, it's probably why it does it. We don't know. We haven't been here to see it happen. So with the virus, I will pass these around. These ones have got it. These are off Dianella longifolius. This is a grandis. I've got a longifolia there. Those two are both grandis. If you know Benambra, I've got the one there that's actually producing the fruit. And these ones, and what it does, so Daniel's not explained, but he did to me years ago. What it does is the flower, the virus or the insect, whatever gets in it, the gall gets into it, makes the flower not open and produce the fruit around the flowering material, and it keeps producing this fruit. So when you collect that, if you cut it open, you'll find all flowering material, and you'll find all the stamens, the whole lot's in there. It hasn't been pollinated. That's why it's doing it. It needs to be pollinated so it produces a fruit, like a tomato does. A tomato's got the fruit inside. It's the same thing. It needs to be pollinated so the seed goes, okay, I can produce more seed inside it, and then grow my fruit around it. It hasn't produced seed. It's got flowering material and it's trying to protect itself. You see, like on citrus gall or the acacia gall we get. The acacia is very similar. It's galling around a fruit that wasn't able to produce seed, and the plant thinks it's trying to still do it, so it keeps producing material around that gall, and that's why you get things like this side on hopefully on longifolia, so we don't get more of that around the place. But I'll pass these around. It was Keck Nursery, I think, wanted to see if anyone here is from there. I'll pass this around wherever Peggy's gone. I'll pass it around. Now, have you finished with those eucalypts? Has everyone seen that eucalypt? Can anyone tell me what it is? Do you know what species it is? No. Sorry, close. So you got goblets, so that's close. The reason I was saying to you is because I want you to understand, because the plants that go with it are the important thing. It's on the ground. It's got flattened peduncles. Well, this one hasn't really, but that one has. They're both Gonia calyx, both. And the reason I picked them up is because I looked at that and went, well, that almost looks like it could be something else, and that's definitely a Gonia calyx. You get the flattened peduncles there. Around these will grow, this is Bundy forest, so this is Hovia, Canetia, Demeter, this type. So it's dry, rocky soil. So they're the species you're looking for when you see a plant like that. You brought up obliqua. When I said, when you see obliqua, best stuff grow around obliqua. Everything that's good grows around obliqua. If you go to obliqua forests, all the good plants are there. In Knox, through Basin, through this area here, back in the 1900s, they've got records of them coming from the city, going to Bayswater Station and looking at these plants, and they're all Gippsland plants, and they would have been around obliquas, because obliqua was right through Old Joe's Creek, up to where, on Dorset Road, all the way around that area, and that was the hot spot of, for me, what I've looked at the maps of Western joining Eastern was actually what we've got through this bloody gully here. Bayswater, the concrete that's on Bayswater is all the rarest stuff of the whole state. It's the most ridiculous place in the world, but if you live in the concrete, it's horrible. But so it all joined here. So seeing a species, like I was saying, with if you're out at Bungaluk, you'll see Cephalocarpa. If you don't know where Bungaluk Reserve is, it's up on off Liverpool Road, and it's one of the prime reserves in this area. Basically, probably is what's left around. It's better than Baton Street, and it's got those two species are dominant, and the plants that are related with them are there. And if you saw that type of plant, those type of tree coverage again, you could probably see the same type of stuff. Where we go somewhere like Ballackwillam, we've got granitic sands, and we've got basically red stringy, bit of Cephalocarpa and Gonia calyx through there. There might be some radiatas down lower on that, but you'll get certain plants up there. There used to be Dilwinia glabrema because it likes to grow on sand. Down my way, I'm there on the beach, I've got sand dune and I've got Dilwinia glabrema growing on it. So it likes sand. So when you see that, you go, oh geez, and then the plants that are associated with it, what else is there and what you can associate, what you might find in these places. Go on to a bit more. We've done the damage, yeah, right. I've talked about that. We're going into processing, cleaning. Look, I said I'm not going to talk much about cleaning. A little bit of that. I could show a little bit later. I could do some eucalypt, show how quick you can do it with if you haven't got sieves. We never had sieves here, and I used to clean seed for bloody hours. Pultoneas, 2 days, 2 nights. I can clean Pultonea with we got crushers and Dilwinia like this. I can use one of them in this. Crush the seed in there, put it through the sieve, bang, bang, bang. I've done it in 15 minutes. Pultonea gunnii. I sat and timed it and it was clean. I go, just using that. Properly ripe seed done instead of 3 days. Just having one of them. We've also got, I don't know if anyone's seen the Seeding for the Future video, DVD, years ago. Has anyone seen it? We have copies still here. I can probably get it played, but I mean, it's an hour long. If we get it copied, I'd really suggest you take it back and maybe have a night at your nursery where everyone gets to see it. Because the methods she does for sieving, winnowing and that type of stuff, which I'm not going to do because it's just too many. I really wanted to do collection and storage for you because I think that's more important than your cleaning. That's totally different. And hoping one of you do that.
Where'd you get the sieve?
The sieves, these sieves come from place, used to be Shore Grow. I don't know what they're called now. Harbour Green? Look, you can get the bigger size, but our seedy people wanted the small. They haven't got the rest of them there. They like this size. I know at Candlebark we had this size and we used to use this. Bursaria. How much are mongrel Bursarias? You can get these and make sure you get the right size and get it. Bang, 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 bang. That one's got seed, that one hasn't. The seed's there. Take the bits out, you're done. 5 minutes. Instead of sitting there trying to pick them out of the pods and that. I've got Bursaria seed here. I can show you. I could even go through showing you out of the pods how quick and easy it is. There's the seed. Done. And of course with Bursaria, is it worth storing? Well, if you get enough of it and use it that season coming up and you've got enough left over, store it as a backup. So most of these Olearia lauratas, Bursaria, any of the fluff stuff, we store. You can have a look a little bit later how we store fluff. We store it away from UV light too. UV breaks down Asteraceae, so because it likes the light. So if you've got it in an open container and sun hits it all the time, broken down. You've lost outside of it. But if you've got it in a bag, at least in a dark area, dark box, at least you're stopping the UV light from hitting it. Sorry, I lost my word. With the sieves, yeah, you can use a certain amount of them. You'll get to know what seed you're using. There's 7 there. I might, if I'm doing something like Canetia or an acacia, I'll take the 2 fine ones out because I don't need the fine ones. The seed's not going to drop through there. But if you're doing something like Stylidium, you'll want the finest one, or you'll want a chicken shaker. As a seedy lady's got, they've got a book. We put little things on, and they know what we got. Little chicken shakers of the chicken salt, and the Stylidium seed goes through, and the bits of gloom don't, and you just get seed, and it's nice and clean. It's only a little thing like that, but it's the fine enough, and it works for them, so they use it. You have got fine enough on this, and you can use it straight through that, or you can do it that way. But I'll take out certain sieves for what I'm doing, because I don't want to have to clean it out of everything. I even clean Cassinias and all those through that. I find putting them in with the biggest one at the top, putting in, shaking it, the seed will fall down to the tray you want. You'll get it, there it is, and then I'll put it into the bottom bowl, and I'll winnow it up. And the seed goes up the top, and all the glooms and the rubbish, and you just pick the seed up and you put the seed in the bag. Some of this is close to that. I don't think I've got one here that I've cleaned 100%, but I've in the past I'll get that much, and it'll be all seed, and you go, well, you haven't done much, but there's probably 500 seeds in there. So there's more than enough for you to do 2 sowings of it. And when it's something like O'Leary or something, you probably don't want more than 2 sowings anyway, do you? You want to know if you're putting in, we're talking about bad seed, you want to know you're getting good germination rate. You've got good seed, okay, I've put in 3 grams, I know in 3 grams per gram is this many seed, I know I'll get. Another thing that hasn't been ever, you know you're going to get that many. Another thing that hasn't been done, is all use that. It's got some information, Murray Ralph's seed book. It's very, very helpful. It's got all the information for eucalypts you ever need, but a lot of the other plants it hasn't done. And what I was talking about a little while earlier was sharing information. What we need to learn is how much seed per gram, and I've done a lot of that work already. We have a list of, I know that there's 135 blah blah seed per gram of that species, so I know if I put 3 grams in, I've got 400 seeds. I've got about, and I know it's got an 80% germination rate on, I'm going to get 300 seeds, or 320. Okay, so if I want to get it right, so but it hasn't got how long seed stores for. None of that tells you. He might say something like Asteraceae, it might be, he's got Divisia, C. aotis, and you go back to aotis, you just boil the water, but how long can you store it for? With going back to Fabiaceae, I haven't talked about much yet, I've found even fairly rubbish seed still will germinate in earlier months. So if you're cleaning Fabiaceae stuff, Hardenbergia or some of that, and you pick out the really good stuff to keep, and you've got this rubbishy stuff left over, sow it. Why not just sow it? Especially if it's something like Hardenbergia or Hovia or one of those rarer plants. Sow it. It's more likely to come up. We did the Platylobum infecundum, which infecundum means doesn't sow, but we got seed, and a lot of it was really weak, tiny stuff, small mat. I mean, it was pinhead side. No way. They came up. So don't give up on 100% on it. Try it. Put it in, especially with any of that Fabiaceae, any of the acacia. It's worth having a go. But you don't need to store it. Something like Acacia stricta. You can see it's not seed. It's flat. Hasn't even got anything in it. There's nothing in there. It's just a casing that nothing grows, or it's been chewed. So when we're cleaning, we're making sure that any chewed seed in there is gotten rid of. And you can tell, you just crush it, it falls, it goes. Myrtifolia is one, it's an easy one to see. You go crush, it's already chewed through, you can see the hole. Make sure your grubs are out there. This is the thing with when we're collecting the collecting bags and we're drying it somewhere. I like to put the bags, especially Fabiaceae, out in the sun, on your veranda, hot 30 degrees hit it. Any grubs in there are cooked. They're like little bits of concrete. They're gone. You've killed them. So you've gotten rid of your problem there just by doing that. That starts the process of opening the seed up if any of it's shut. And then the seed cleaners, whoever's cleaning it, you'll clean it. And then sorry. I don't think freezing is the way to go with seed. I think freezing would wreck seed. It bursts the cells on things. I think that's why they probably use liquid ice, don't they? They use that instead. I don't think they freeze. If you're going with fridging seed, okay, any seed, and we don't even know, I mean, you could have Olearia lorata store it properly, clamp it up, put it in the fridge, and in 6 years' time it comes up just as good the first year. Don't even know that. We don't know that. They're the type of things we need. This is the interaction I want back from you, and we want as a nursery. We want you to have a go at this, because this is what we've started to do. We're writing down how long. Yeah, you're right.
I've worked recently with Green Australia and Community Nurseries too. We did a sweet seed swap with them.
Yep.
Because they wanted certain species that we had. They've actually got a list of how many plants for each gram of seed.
Yeah, have you got, look, we've done a fair bit of it, but a lot of species I've never got around to doing. I mean, we even did good here. And the bloke I had collected, he got about half a gram, and he goes, there's about 800, and I don't want to keep going. I say, okay, we'll call it 1,600. But he did 800. He was pretty keen. He did, but we got proper. We looked at what we did is when we counted, we got seed that was fairly good looking seed, like good seed. I want to count good seed. I don't want to count. If you've got Hardenberger, it's like that. It's good. If you've got Hardenberger, it's half the size. I'm not counting that as a seed because it isn't. You want to collect good stuff. Yeah, but if you have, look, if any of that information get around, I can give out what we've got. I've been working my. Yeah. So I know what Pultonayas are about 250, or I think it's 325. It might be for Pultonaya gunnii. And just knowing that, okay, I've got through. I put it. For me, it always used to be 1.5 grams because I knew I'd get 400 plants. And whenever, again, I'm sure you might have been told this. I remember went to years ago. We went to a session out at Nillumbik, and they did a cutting session. And the one thing she said there that's always stuck to me, and I'll make sure this happens: when you grow plants, make sure you have enough to grow how many boxes you got. If you do them in boxes, you do 50. You want 100. You don't want 43. You want full boxes. So when you go and put them in sales, I've got one really good looking one, and I've got my backup ones. Or they're the same. When these are half, so I cut them back, I change them over. You want to grow enough that gives you enough to, so unless you're growing rare plants to build up numbers, obviously. If you've got 10 seeds of a plant that's rare as hell left, I'd suggest putting half in and see if you can get a couple up and then build your numbers up rather than chucking the whole lot in and just throwing the whole lot in. If something goes wrong, you've got another chance. If you haven't, you're gone. So make sure you're doing that too with the seed numbers, what you're sowing. I know we didn't get the sowing, but what the numbers you're sowing, if you know the seed numbers, you can do that. And per grams too. Your containers you've got your list. He hasn't got it up anymore, but if you all got a seed bank list, I don't know if everyone does that. I know Candlebark does because that's half our stuff from there. We got some from Crisp as well, some of the ideas from there. But we run one, and knowing that you've got 6.5 grams of Hardenberger and you know 65 seeds per gram, I know there's nearly 400 seeds in there when I sow it. Yeah, Jenny.
With the seed, when you put them for doing the wet heat, the seed that floats, it stays afloat, and I sort of thought, well, that's usually the small white stuff, and that's not going to grow.
It usually floats because it's probably been eaten. It may have been chewed on by an insect, and there's air in it. That's why.
But that, and if you start seeing a hell of a lot of it sort of floating, not sinking, that means. Yeah, but I don't think it's 100% that they are not going to germinate. I think if you're really important, I know we're not doing cleaning on this because it's too much, but it's really important that people who are cleaning seed make sure that the seed they're cleaning is actual seed. Because us growers, the people who are growing it, it's a real pain in the butt if you sow it and you look at it and go, especially if it's wrong. I mean, we've got a couple of wrong this year going, oh, that's not Hardenberger, that's Acacia, I think it's Stricter or something. So make sure whoever's doing it, and this is why it's got to be proper, and this is why you're putting signs in there. That's why you're putting bits of the plant in there. If you're collecting Acacia, put a leaf in there. If you're collecting Pycnantha, put a link that you've got the seed pods in, chuck a leaf in as well. If you're not 100%, if you're not 100% sure, like this, like the Gonia calyx, if you collect that, and I will get up to those, if you're collecting that, you'll put bits of it, even if you put one of those leaves in. Look how long the leaf is. It's a long leaf box. Oh, hey, look, geez, it's got a big leaf. Is it mountain grey or is it Gonia calyx? Because they're closely related. And then the way you tell them apart, one's got the flattened peduncle and one's got the rounded. So as long as you've got all that material in, you put those in there. We even now with eucalypts, we put in the pods with the peduncle in our container because we've had collections that have gone in the past that someone's collected and put the wrong name on and they've cleaned it and put it in there. That's not that. It doesn't exist at that reserve, or that's the wrong time of year, or we've sown it going, they came up something totally different.
So that's another reason you don't want to just blame these people who collect it and get it wrong. You want to be able to ask them, did you collect it from here? How many there were? All those things. Okay, we'll get this out of the way. We'll go on to some. I don't want to put them on there. I'll get killed by the seedy ladies. Okay, we'll go. Sorry. Yep.
Because I went through an exercise about 10 years ago looking at how many grams you'd need and what seed you need there. And the sources that I found from Leon Osman had it for use.
Yeah.
But it's very patchy. There's not.
Yeah, no, that's what I'm saying. Even with this, if all the information's there for eucalypt. If you want to do eucalypt, it's there because they were obviously they were doing all the tests of them. What were going to be the forestry ones? That's why all this information's there. It's only reason, and we don't have that information for all these other plants that we're losing, and we're going to lose them. Something like Thelonema. How do you grow it? Because I've collected it. I've collected it fresh. I've sown it fresh. Definitely been ripe. I watched the blue banded bees pollinate it, and I collected that seed. It's got to be ripe. It's got to be right. It's there, sitting there right there. I've sown it ripe. I've sandpapered. I've heated. I've done all these things to try and get it up. All that. I mean, we're going to get Liam here. You're in trouble here, Liam. Liam's been experimenting with charcoal and ash. And when we do, I know we can have vermiculite that's impregnated with smoke water, but the ash and the charcoal slowly release it like slow fertiliser, and that's what happens in nature. When the plants come up in nature, the colonisers come up first. It's not, you don't see Pulteney Gunnii come up first, you see Solanum come up or whatever up there. My way, I've got Vescums. It's funny, on the beach, I've got Vescum which grows up in the mountains, it's on the beach. And they've disturbed this area and they're all popping up. So it's the coloniser, it's the first one before anything in there is going to come up. And that soil gets heated, it's there and it knows when to come up. But that ash layer it's on there, how long does that take to actually permeate into it and get into that seed so it actually tells it? Because we pour this smoke water over it, it's one. Then it gets watered and diluted out, it's gone. It's still not penetrating into the seed. Vermiculite with smoke water infused into it seems to do fairly well, but I'd be more with Liam with the ash and the charcoal. Please keep experimenting with that. He's been experimenting with when he gets Lepidosperma. Lepidosperma is a bugger. You've got to go through almost through a chicken's butt. But you do I put it in winter, let it wet, then you've got to put it out in summer and let it bake for a season, then bring it back in and do it again. When I first started at Candlebark, Tim Flame, who used to be there, was doing that. And he had this big frame at the back, a cold frame, that took up stacks of room, and there was about 20 trays in there. And there were 6 Lepidospermas in that one, 5 in that one. They just come up now and again. I just go, well, I need this space to grow Pacris and that, so I gave up with it at that stage. We really need a better way than that. We really need to be growing 200-300. I mean, Gunnii seems to germinate fairly well if you get the ripe seed, but it's Lateral and those other ones that don't seem to do very well at all. And those secrets are what we're trying to unlock here, and we're trying to spread. Whether the storage of it for 10 years makes it come up. What are you up to? You want to
Morning
10. Right, There's 3 people still on stools. I can see 2 on the ground. Okay, so look, we'll go on to some of the seed here when collecting time is. I've got some Hakea from out the front there. This is Aulacina. And I've got a green pod there. That's this year's flowering. In the plant, you can see last year's growth is there to there, and there last year's pods on them. This is this season's growth here. The flowers were on it. It's only produced the 1 green pod this year that's there on the new growth there. They're not collectible until next year when this thing's flowering. So I collect Hakea in August, September, not only because it's flowering and you know the seed's definitely been there for a year and it'll be ripe. You can see the damn things, especially in the dosa. You want to find Hakea in the dosa out in some of these bushland? No way. But when it's flowering, there it is. And the seed's been on there for a year from the last year's, you know it's going to be ripe, so you can collect it then. So that's the timing to go. Same goes with Banksias, that time of year, when they're flowering again. I know Spinulosa can sporadically flower throughout the year, but it's around that time of year when you're not collecting other things that you can go. And when I was at the start of this talk, I was talking about recceing and surveying and looking at things. Why you're doing it, that's what you're doing when you're out there. So you're out to reserve, you collect, you go, I didn't know that plant was there, or that was there. We went out to Ballyquillam and I went looking for Haeculacina and I found Acacia brownie absolutely everywhere. I never knew it was there. I've been going, wards. I've been going to wards for nearly 30 years. Before the horsey people wrecked it. It used to have a Pacris everywhere and they used to mow it this high and it was beautiful. Now it's not there anymore, unfortunately. But brownie eye used to be everywhere as well, but I didn't know it was in these sections. I never knew. And I never walked that far, lazy me. I just go into that bit and walk into where the wombat hole is and grab a little bits of and out again, make sure you're, again, not trampling everything, going around, don't fall in the wombat hole. But that type of thing, just by looking for something else, you might discover something else in your area that hasn't been collected from that can add to the gene pool of your plants. And that's the main thing we're aiming here with anything we're collecting. I was going to go through that as well. What you're collecting, what you need. What you need every year. You probably need things like Olerias, maybe Cassinias, you want Dianellas, Lamandras maybe. So they're high on your list when you're collecting those. So they'll be high on your list and when they're collected. So I was saying Oleria laratas now, that's what you go and prioritise. I've got some backup from last year I know germinated. It's easy to collect. Simple one, really. That's probably one of the easiest, as far as I'm concerned, it's the easiest fuzz one to collect other than helichrysums. I'll always call them helichrysum. That's the yellow sun. They're not, someone else can have their name. They're helichrysums. When they get changed back to helichrysum, like Dianellas got changed back to Revoluta, yeah, we'll do that. So we'll talk about that as well. But when you're collecting this stuff, really easy this time of year, and you're going through the process of what's ready at that time of year. What you're collecting, need every year. The priority, what's your rare stuff? What stuff haven't you collected before? What do you want to get? You go, we really want to do Hovia this year. It's a really good for you. And what's the year like? Like this wrecking will tell you that it's a magnificent year for Acacia stictifila this year. That's what we're going for. Last year was crap, so we gave it away. So we went, looked last year, it's not that good. Let's do something else. Let's leave it and try next year when it's good. And we got younger plants here. We know that next year they'll be ready. I mean, I went up towards, I was actually looking for the other Hakea terretifolia, and I'd seen younger ones in there. Of course, I couldn't see them this year, and they got up this year and they had good seed on them. They never found them, but anyway, I'll probably walk straight past them as you do. But doing the recce in there beforehand, I noticed them. So that's why you're doing recces. It's not only for the stuff you're looking for now, like you're going to collect, Oleria laratas, you might see something else that you don't go in at that time of year. Yesterday I went to Hanson Track and walked down. I don't think I've ever been down that part in November. I'd always go on there later in December and go collect seed. I walked there and went, I never knew Poa siberiana's in there. I've never seen them there before. It's bloody everywhere. And we've always been, where are we going to get Poa siberiana from? There it is, on that basin side of the hill where it should be, running down like it is in Bungaluk right through to Croydon and that area where it grows out to Mount Evelyn. So just going in, looking at other things, doing reckings, you'll find stuff to go, damn, I know. And recording it, make sure you're writing this stuff down. Unless you've got a steel trap memory, I know where everything is. I don't usually write it down, it's up here and I have to, it's there, yeah, it's there. But if you don't, you want to keep a list of where things are. We have a collection list, which I haven't talked about yet. Maybe Richard will put it up later so you can see. I do every year for them. And what I'll do is I'll do all the species we can collect or we want to collect. And ones that I go, oh, well, look, we don't need Acacia stricta. I'm not even going to put that on the list this year. I don't want anyone collecting it. Or, yeah.
Now, Woods Road.
Yep.
Here we went collecting out there.
Yep.
And we saw bags, must have been on the Hovias. And we wondered a little what.
That'll be candlebark.
Right, so that brings up the question of who's collecting where?
Yep.
Are we intersecting over
Overlapping. Yep.
on the same basis?
Yep.
Because my bunch, we go out towards Road.
Yep.
Yeah, the brownie eyes faded a lot.
They sprayed them, I got told. On the edge there. I had another bloke, he's been going for 30 years, said they sprayed them. But they're down further, down near the fence, down the other side, there's a really good population.
Yeah.
It's not about 1 year the Hovias were bagged. Yeah. I can guarantee you Candlebark hasn't collected there again, so that's...
No, but it's more about that intersection of who's collecting
yeah.
That's my point, because...
And normally there'll be a label on it who it is. We always labelled anything we bagged as well with a Candlebark collection, or we do here at KS2, so I'll normally have a label run around one of the plants that says, but you have to find the one.
It's that coordination
Yep.
between the various groups.
Yeah, no, I totally agree.
Like wards is probably the most northern we go to. And I suppose from a seed collecting perspective, you don't want to have too many.
Two people hitting the same spot.
The same spot, because if you're going, oh, I get 10%, and if somebody else has already been through on their 10%, and another group, another 10%, you're really hammering it.
Okay, you might get a bit up. Richard and I, we're total advocates for this. This 10% thing going every year is ridiculous.
Right.
If I go on a good year and I get an amount of seed that I need, the amount that I need, the Aboriginal people, they took what they needed. They didn't take extra. So if there's 1,000 seeds there and I take 500 and just leave the rest because it's a really good year, who cares? And then I don't go back there for 5, 10. But our real aim here is actually to grow the plants here so we can collect the seed and those gene pools that add into what we're doing. Not like the old days where it was everywhere and you just collected and just did it. We know that is totally different now. What you're collecting is to build up your gene pool so you can regrow them. This is what this is for. I brought these out for. Growing at your nursery is much easier. Look at the Canetti. I know I've got my Oxalis, that's from my one from home. It's very happy with the other plants growing with it. When they grow by themselves, by the way, Canetti, they seem to struggle. When they've got other weeds and things growing through them, they grow really well. They really like it. So I just leave it in. I just run it around a pot. But our aim is actually to get the seed stored away so we can do those things and have enough so we can do it. It's not, let's get a whole heap, make lots of dollars and then go back and store it. These are not, we're getting, I know we're getting accused of this. There's people selling online that are doing this. Yeah. Definitely. And definitely some of these bigger nurseries who are basically our enemies. If all us here are community nurseries, the big nurseries, I won't mention the one out here in Wanturna. I'll just mention it's in Wanturna. But Bushland Flora, those places, they are competitors with us. And they have, in their nursery, they'll have 12 of me who know how to collect, how to grow, where us little nurseries, if you've got one of me and 3 other people that have a go, you can't grow the plants as well as these people who just pump them out. And these big, that's what they've got. They've got experienced people who do this. They're the ones who are really pressuring us to keep us going with your orders. But I mean, if all you nurseries aren't focusing on retail, you're silly. Because retail is the market, seriously. The people out here want these things. They want, I grow wormbia, really easy to grow. Once you know what you're doing and once you know the process with it. People come and they'll pay the proper dollar for it. You'll get your money to do your whatever projects you want to do from these rarer plants, and then you can do this other stuff. You can grow your 1,000 eucalypts a year, get 20 planted, who cares? They turn into mature trees, chuck the rest. If you told me I grew 1,000 plants and 20 turned into mature trees in Knox, I'd say that's a success. I'd say every single mature tree here is worth more than those 1,000 tubes. Yeah. So the big, beautiful yellow box we got out here are better than the tubes. So probably got morning tea. Do you want me to have a rest?
Yes. So we'll have a.
If you haven't seen, I've got breathing apparatus going up. Haven't stopped. We haven't seen him.
We'll have a break for about half an hour. There's tea and coffee and everything like that. If anybody wants a tour around the nursery, if you haven't been before, happy to run a tour.
I'm only going to do about 10 or 15 minutes more. We want to do some bagging. But on that seed collecting of things that you want to get the whole of, if you've got roadside vegetation, anything that's on the edge of areas where seed's dropping that it's never going to germinate, go for it. Don't feel guilty, I better leave this. As I said, it ends up getting mown, it ends up getting smashed. As Vic Roads keep deciding they're going to go further up banks and further in, they're taking out all this really good plants. Years ago in Yarra Ranges used to have posters and things, how proud they were of their roadside vegetation. It used to be like a big thing. Now you go out there, it's terrible, it's gone. And all you've got on there is weeds. And weeds, as you know, burn. When they talk about grass fires, they're pasture grass fires, because pasture grass has nitrogen in it, our native grasses don't. You might as well make a bomb, they're bombs. So if you got that stuff on edges that you got a chance to get, do it. Go after that stuff, get it. There's a couple of places up where I used to collect up near Seville. There's and Divisia latifolius here in Knox, it does it too. Usually on a slope on the edge of the road. That seed's only dropping in the gutter. It's never going to germinate. The pH of the soil and the makeup of the soil is not what it wants anymore. Most of our seed drops and the soil's set up for it. That top layer of that really silty soil, that's the germination layer. That's like sowing mix. That's like seed mix, that. And the clay that goes through down to the clay layer where it gets its moisture from. That's how our natives grow. They don't get watered from above. They take water from the lower clay levels where water used to permeate down from the higher levels with the hydrology we don't have anymore. So I know it's pretty complicated to think about, but that's where we're at. That's why we're losing a lot of our plants. We're not losing them because it's climate change. That's a crock of shit. The reason we're losing them is because the hydrology in these reserves has changed, and the water level has dropped so far these plants can't reach that water anymore at that time of year they need them. So if you're getting that type of what's happening, you've got to get those plants out and you've got to find a new place for them, because that's what would have originally happened here. If somewhere turned, it's now an inland sea, those plants have got to move, or it was an inland sea, it's gone out like Victoria was, the plants move in. So it's there to do. Now what I really wanted to talk about was, I've still got a couple of eucs to look at. I want to show you with eucalypt. We saw those ones, just what were in a reserve. When you're collecting eucalypt, what you want to collect, this is off a yellow box. These green ones aren't any good. What you're looking for is the older material, older seed up higher. So it's flowering like we did with the hake, it's flowering at the moment. So the old growth, the old last season's growth, there to there, that's probably the season before, that's probably one season, that's probably another season. It doesn't look like it produced seed that season, but it did that season. So you're looking for older seed, like this was on an old, that they haven't opened yet. Some of them might be actually blind, but they've changed colour. They're way back on the stem where the growth is, so that the season's growth are there up to there, and they're back here. That's gonna, when you dry that, you're gonna be able to store it. If you could, this green stuff, you could probably collect it. It's still in flowering now, but if you collected the stuff that's stopped, if you collected that now, it might dry out. You might get some seed out of it, but definitely won't store. It doesn't. I was hoping to have Max here, our eucalypt expert, to go on a bit about that, because this is what he does. He goes all over New South Wales and Victoria collecting eucs, and he points this out all the time, and you can see the difference. You can see where those green ones, and you're aiming for those ones a little bit higher. And when you're collecting eucalypt, if you've got an area that you know always produces good seed, don't cut the whole branch off. Leave the branch hang down, take bits of it that produce it. You keep collecting from the same spot. You don't want to be always having to climb a tree or using a huge pole to get this stuff. So have a look at that bit later. I want to do a little bit of bagging. I'm going to use pantyhose. Richard's had heaps of these in his car for some reason. I do as well, but with
Smooth and silky.
yeah, smooth and silky. Yeah, they're really good. With the Viciolatifolia, the Hovia, anything that's got a long stem, you want it using the bags. And when you're using bags, so you want to use the lighter colours. The problem with bags, as I was saying before, with them, but bagging things too early, what you're doing is you're stopping the solar radiation onto the seed that heats the seed up, that actually makes it ripen. It's like an apple. The apple sits in the sun. The reason the skin gets harder is because it ripens outside, because it hardens. And that's what helps it. That's why the sun hits it. If you put stuff over it, you put bags over it, it wouldn't. And the same goes with this. As I said earlier on, I bagged Hovia years ago, really early, and it didn't go in it. So when you're doing Hovia, it has to have changed colour. Same with I'll do that later. But with the Viciolatifolia, what are you looking for before you do? These are too sorry, these are too young. You can't see any seed in the actual pods. So when you're going to do this, you want more pods than this, obviously, but this is all we've got here. We've got more out in some of our reserves, we've got more. But you're looking for the seed in there, I can see one in there, through there, before you even do it. And when you can see that seed, that's good enough to do the... This, by the way, this isn't, you can come and have a look at this later, you cannot see the seed on these. These aren't ready to do, so I would normally avoid this and I'll come back in a week or two. But this is what, week or two time, this is what you would do to do it, and then you just tie it off with a twist tie or like that. The seed drops down there, you come back in late December, mid-December, the seed's all in there. Cut the plant back, they love being cut back. They're roses. They love growing from old. This needs to be cut back down there, so next year it puts a new shoot, and that's what it flowers. And you can see it's flowered like a fruit tree does on this new growth. It hasn't flowered on old woody stuff, it's put new stuff out. Next year, this one got cut back, it's going to put fruit on it. like apples and that do on that second year growth. So you want to, if you've got one of these in your nursery, if you cut it back on a regular basis after they're flowered, so cutting it there, you will get induce the plant to produce more seed. Yes?
Can you cut it back into bare wood?
You can if it's got buds in there. I think I would actually cut it down to that newer one there, that green one, and cut this greener one back if I was doing that. Then if a new one produced down there, I'd cut down there. If you've got access to ABC, if you go back to one of oh God, I've lost his name now. I should know. Sorry. No, better than that. Peter Cundall. Better than that. You'll go and see one of his rows when he does espaliating or any rose pruning. Watch him do rose pruning. If you want to learn how to prune properly. And these are basically a rose. If you cut back like that to new green wood that's coming up, you'll produce masses of seed every year. That's what they want. What happens to them in nature, the animals would have come along and chewed on them, chewed the new shoots off, and that's what they want, like Pacris does. Pacris sends out new shoots around its flowers this time of year. They're hoping the animals eat them and then go away, and then it shoots the next ones out and they've got the flowers on them. So cuttings this time of year, I know we're not going about cutting, but cuttings are the ones around the flowers because they're the ones that are going to put another growth on them. You don't want to take the next growth because that's got flowers on it and you're taking seed from the bush. If you're taking cuttings at the right time of year, you're actually helping because the animals just aren't there. Like I was saying, I was in up in the national park here. There's not wallabies up there. There's deer, but I don't see, there's not many wallabies anymore. And the wallabies are the ones that do the browsing and do this job. They'd be up that height and they would tickle these down, like the leptophilus up there. They would do that this time of year, give them the little tip prune and they'd reshoot out. With using the gift bags, if you do these, onto the, I do Canetia, sorry about with Canetia, which I said, this is my little beauty at home with all the other weeds. I would get it if I was out in nature, out there somewhere. I would just put him in like that. Only small batches. You don't want to put too much in. If anyone does Hybertias, when you bag Hybertia, you only want to bag one branch in each bag. Don't try and put crap loads in. Oh, I'll stick more and I'll get more seed. I did that years ago, and I did one with one bag and I got 10 seeds, and I did one with about 5 in this bigger one, and I got about 3 seeds. I just keep running around and it'll produce as much as you ever wanted. With a couple others as well. Remember, when you're doing any of these things, I always have 2 or 3 provenances next to each other, so they've got different parentage. So the cross-pollination is actually not inbreeding. Because you could get 3 from the same place, it could be from the same parent, and you're actually only inbreeding. You're not actually going forward, you're actually going backwards. You're going back to Tutankhamun, you go back to his dad. You do his dad, you don't want Tutankhamun, and then we've got to take cuttings, I don't want to go that far back. So bagging like that, I do that around late November, a lot of our plants, the hovias, because they're ready about that time. It's usually mid-December we'll see seed on hovias. It's changing that drops, but it's late November, early December that they change to the darker colour. Same with them, they start to go that peanut, like they get that little bit of hair on them. When they're ripe, they'll be like a peanut. it's like wood. You can click it straight open like that, and they'll just split and the black seed comes straight out. If you get some seed that's a little bit greenish, especially with hardened burgess that's not properly ripened, I've put an Acacia myrtifolias one, because it always just bursts. I better get some. It can be collected a little bit green, and you can ripen it up in the sun, and it will change that olive green from the light green if you collect it a little bit early. Because not all of it's green properly ripened when you collect it. You always get a bit that's, and you dried it out, and you put it in the bag, left it out in the sun, it's all burst, and you get it, and some are a little bit light green. Get it all in a container, put it out in the hot sun for a couple of hours, bang, they'll change the olive green. So does hardened burgess will do it too. And Canetia will go black if it's put out. If it looks a little bit, it's not quite there. So, but again, if you're going on a regular basis and checking up on these plants, these rarer ones, this is the ones you're really trying to do this for, you'll get that. And then going into pot culture is, for my advice, is the best way to go. Look, we've got, this is Acacia aclatissima, as I was saying. We get seed on it when we grow it here. Sorry. But when we grow it, when it grows out, and I was saying, when it grows out in the wild, the seed is far and few between. I've been out collecting 2 hours and got half a gram of seed of it. We were desperate for it, and it's not worth your while. Whereas if you can get it growing into pot culture, you can do it. It's the same as a lot of these plants. If they go in pot culture, Pacris, you can collect the seed on site. The cuttings strike a lot easier because they're grown in potting mix. So everything points to doing this if you're not at the moment. And overhead watering is the killer. I mean, if you've had a session with Daniel, he would tell you this every time. Who's the one on about it? You cannot overhead water your plants you're collecting seed off. It just rots them. Now you got to try and imitate what's in nature. I went through the Mallory Ralph book. We went through the time that seed stores for. We was talking about that before. I think I'm just about finished. Is there any other questions? I'm sure there's crap loads.
Just a quick question. Those little seed bags, what do you get those?
They're just gift bags. You buy them at the $2 shops. They come in those little packets and you get about 10 of them. Okay. If you've got a mixture of this size is pretty good. The little size, it works for a couple of things. There's a couple of plants that it's really good for, but it's probably useless. This size and the medium size that I put on that one are the one. And if you can get the lighter colours, you want white or yellow. Don't try not to use red or any of the colours that are part of the spectrum. Arthur Daly's, yeah.
So thanks a lot to Jamin, the great man that he is. He's full of it, and that word is seed.
Thank you for that.
Yeah, thank you very much for your time and effort. We really appreciate it, and I'm sure you'll see him again because he's a valuable resource. But if anyone else has a valuable resource who's willing to give a session, then it'd be really, really good. So there was a thing last year, someone wanted something on wetland plants. So if anyone has a wetland plant expert, they'd be willing to give a session, just let me know and we'll see if we can organise it. Otherwise, thank you very much. Thanks for coming. Still plenty of food and drink, so make the most of it. And we'll see you next time.